Thank you, Alberto and Terry, for organizing this very nice program. And I'm happy to give some talk about some aspect of interface growth based on this experiment that was performed now more than 2 decades ago, where they put a drop of bacteria that were kind of identical, except that they were engineered to fluoresce in 2 different colors of red and green. So this initial drop that was a mixture of red and green bacteria was placed in the middle of a growth medium, and then the bacteria started reproducing and expanding away from the center. And what you see is that after a while, there are sectors that are one color, green or red. And the reason for that is that basically when you have a lot of green, all of the children will be green in color. Where you have a lot of red, all of the children will be red in color, and red will not suddenly appear in the middle of green and vice versa. So this is this kind of sectoring that takes place. So then what they said is, let's look at the boundary between 2 of these sectors that are red and green. And what they found was that the fluctuations of this boundary, that is, you kind of think of it as a path that is progressing, and you may think, okay, it's a path that is progressing. It can go up or down because of the way that the random reproduction is taking place, and so it should be like a random walk. And so if I go a distance x in this direction, fluctuations in the other direction should be of the order of square root of that. Okay, so they did the measurement, and they found that the mean squared fluctuations, rather than scaling linearly with the distance over which you are traveling, were scaling superlinearly or super diffusively, if you like, with an exponent that is close to 4/3. Okay, so they also made a very nice model. And so basically the idea of the model is that you have these cells, and here they're placed on the sides of a lattice. And at the front, where there is open space, the cells can reproduce. And if you have reached a level where there is a red and, let's say, blue that are on the front, one of them randomly will put its children over here. And because of that random placement of the child from red or blue, the boundary, the sector boundary between red and blue, will fluctuate diffusively, and you would expect this t to the 1/2 behavior, which is not what they saw. So the next version of the model, they just made a simple change, which is that in this first model, the assumption was that you grow one layer, and then another layer, and then another layer. And so the interface between the growth and the empty is flat, which is certainly not the case, because the cells don't wait to make sure that the entire layer is filled before they reproduce. So the other version of the model that they had was that any cell that was on the boundary could reproduce. And then again, there was a boundary between 2 different colors, and now the fluctuations of that boundary had this 2/3 scaling, which was seen in the experiment. Okay.
So these are results of simulation, or we were able to prove?
Well, this is the result.
The 2 models.
The 2 models. Well, this model is obvious. Okay.
Yes.
This model is one of those growth models, ballistic deposition, etc., that was mentioned already in the. So if you believe that the interface of one of these models with one color has these fluctuations that are characteristic of KPZ, then I will prove for you that the boundary will have the super diffusive scale. So basically, now let's sort of zoom out and think about things at a larger scale. let's imagine that at some point in time, our boundary has fluctuated, and locally it looks like it has some particular slope. So the height as a function of position, if you look at the boundary, is not flat, it is at some slope. The slope is, of course, dh by dx locally. Now suppose that we are looking at a portion of the boundary which is really the sector boundary between the two different colors. Let's make a very simple assumption that each color grows normal to its current position on the interface with some velocity v, so that after a time v times dt, the boundary has shifted from what is here to what is here. Now what you can see that from this simple geometric construction is that if I look at the x location of the boundary, it was here, and now it has moved here, and the drift is simply proportional to the slope times this average velocity. So essentially, if you are at a sloped surface and you are just growing normally without thinking anything about fluctuations, etc., you will be drifting one way or the other, like there is a force along the slope that is sliding you one direction or the other direction. Okay? Now, since the whole surface is randomly going up and down, if you look at a particular point, the slope is also changing as a function of time. So this again looks like a random walk. You have essentially the position changing randomly, but the amount that it is changing is through this slope, which is a correlated random variable as a function of time. The value of the slope at some time and some later time is going to be correlated to each other. So if we can figure out something about correlation of the slope, we can integrate that and get something about the root mean square fluctuations. Okay? Now the same construction that told you about this drift of the interface also tells you that the height fluctuations, the height growth on a sloped surface is proportional from this geometric construction to the square root of 1 plus grad h squared. You sort of have to jump more over here, and this was already mentioned by Kaz. And so if I expand that, I will get this nonlinearity of the KPZ equation. So if I want to sort of figure out what the slope fluctuations are, I have to look at this equation, its noisy variant, and calculate what the scaling of the fluctuations of the slope are that is determined by that equation. Now there is a lot known about the scaling of the height, and if you take its derivative of the slope of this equation, some of it was again mentioned today, and essentially what happens is at the end of the day, root mean square fluctuations of the boundary inherit the dynamic scaling of the fluctuations of the height of the interface. And so since the height fluctuations and the dynamics was governed not by a diffusive exponent z, which would have been 2 and would have given us the usual x squared goes like t, but by z of 3/2, we get this super diffusive scaling that was also observed in the experiments. Any questions? Okay. Now, in this experiment, it was very precisely engineered so that the two bacteria that were growing were essentially identical. They were growing with the same rate. But typically, if you have normal bacteria that have different origins or properties, typically one of them is more powerful than the other one and will displace the other one. And the language that is used is typically fitness, that in this case, the bacteria that were in the center were more fit than the bacteria that are colored yellow. And as a result, they started with a small seed of these dark ones, and this seed grew and grew and grew and displaced the yellow one, and ultimately the yellow one would disappear completely. But then as a result, also the shape of the front has this characteristic bulge that will not surprise you, that should be there. So can we describe these shapes and what they could be? So I already told you how the height of the front is changing. There is an average velocity, there is this nonlinearity, etc. But now we have another variable to consider, which is that when I look at the front of the interface, it is characterized not only by its height, but whether the bacteria that are at the front are of one type or the other. So let me introduce this function f that is 1 when I have this new, more powerful bacteria, and is 0 when I don't. I have the other one. And so if I look at this interface over here, I have this one-dimensional function that is 0, then becomes 1, and then goes to 1. And if I look at this function as a function of time, clearly it is something that is expanding. So essentially, the shape of this function f should be described by something that is expanding. And what do we know about equations that describe something that is expanding? It's the well-known Fisher-Kolmogorov type of equation, which says that essentially the f function has two stable points, that is, that has 2 points, 0 and 1, that it can be stationary. One of them is stable, one of them is unstable. And then the front has some kind of a diffusion, which is reflection of the fact that the children can be placed to the right or to the left with equal probability. So in the continuum, you have something like the Laplacian. So if I start with the black terms for f, then I have just a front that is expanding. Now I can ask, what is the simplest way to couple these two equations? Well, one term is what I have indicated here, alpha f. Essentially, it says that when f is equal to 1, the height is growing more rapidly, which is what we are seeing over here, compared to when f equals to 0. So alpha is really the relative vertical growth rate between these two. The other coupling that I have is exactly what I had told you before. That is, when you are on a sloped surface, the boundary is going to slide one way or the other, and the effect of advection with a velocity that is proportional to gradient of h is simply indicated by this term. So this is really just f being sliding along with h. And for reasons that will become later on clear, actually there is really, for thinking about morphologies, etc., I don't need to include noise at this stage. Any questions? Yes?
So I guess, so the coupling alpha f, somehow I would think that, if the bacteria are just, they're dividing, then it matters whether they're on a sloped interface or not, and how they affect the height. So should there be like a gradient coupling?
Yes. So these are the lowest order powers in f and h that couple together. But really, I should not restrict myself. I could, in principle, include higher order powers of f here, gradients of f, and things like that. That's true, yes. In fact, it should really have an f gradient of h squared term with alpha also, but it's higher order in this. Yeah?
So the term, the advection term, is kind of, so for a given slope, graph h is pushing more the crop?
Yes, it's sliding So if you are on a sloped boundary, the location of the transition point between 1 and 0 is going to be pushed to more. Yes. It's just the same mechanism that I had given. Okay. Now, what are potential solutions to these equations? One solution that kind of looks like this is you would say that the blue that want to grow with velocity v0 plus alpha will just bud out and form a semicircle or part of a circle that is growing with velocity v0 plus alpha. The other part grows just linearly v0t, and I just have to patch those solutions together. So that's one possible morphology. If that is the case, then the boundary between the blue and black in the horizontal direction will be moving to the right with a velocity u, which you can simply get by geometric construction from square root of v0 plus alpha squared minus v0 squared, which to lowest order is just this square root function. Okay, that's one type of solution. Now, since we started thinking about Fisher equation, and the characteristic of Fisher equation is that it has traveling wave solutions. That is, you have a solution that depends on x and t through the combination x minus ut, which is essentially translating that whole thing. So we can ask whether our solutions admit something that is of the form some function of x minus ut. And if I try this function, essentially the d by dt parts of my equation will get a function minus u, and the derivative, and essentially I will get an equation from which I can solve for what the derivative is of the function. And then ultimately, when I look at everything, it turns out that it tells me that the boundary between blue and black comes down with a slope, and this sloped interface is simply translating. Okay? And the speed and the slope are related to each other through this nice formula that, again, solving just quadratic equations, etc., immediately tells you that this relation takes place. And so basically, this is a different type of morphology. Perhaps you say it's not so, you really need something precise to see whether at the edge you come with a slope or you are part of a semicircle. How can you tell the difference? Well, the thing is, however, that the value of this slope can be changed from positive to negative, and that's an acceptable solution that looks very different, where this is the circumstance here where the parameter that I call alpha, rather than being positive, that was pushing up the blue, is negative, and then I get this kind of structure. Now, from the perspective of mathematics, this is trivial, but biologists maybe have a little bit of difficulty with this solution. Why is that? Because the typical way to see whether two bacteria, which one of them is more fit than the other, is that you separately grow them on the growth medium, and you see which one of them is growing more rapidly. And you say, oh, this one is growing more rapidly, black compared to blue. So if I were to put black and blue together, then blue will disappear and black will take its place because it's growing more rapidly. What this shows is that no, there are circumstances in which the growth rate of blue is less, so it would grow to a smaller amount compared to black, but the blue can push out the black and ultimately be the winner in the competition between the two, but through this new morphology. Yes?
Without pushing out the black,
So if blue bacteria or bugs, if they die quicker than the black, this can happen, right?
Then there is no reason for them to push into the black if they are dying, right? So, they reproduce slower, but in competition they are, yes. So if you like, one way of imagining it is that these are really tall guys, these are short guys, but in the battle the short guys can beat up on the tall guys. All right? So height is not everything. Okay. Now, as I said, this was maybe a little bit of surprise, except that only about, I don't know, 6 or 7 years ago, in the lab of my colleague Jeff Gore, they found an example of this kind of system where you have these real green bacteria that are expanding into the red, but at a slower rate in the other direction, and the morphology of the front is something like this V-shaped type of morphology. And so basically, as the parameter alpha that shows the relative growth of these two species changes from negative to positive to larger positive, you have some kind of a phase diagram in morphologies that are governed by this pair of equations. Okay? Let's see. All right. So usually I skip the next two transparencies when I'm talking to physics audience, but since there are mathematicians here, okay, let's go and have some equations. So what I told you was that there was a relationship between the slope and the speed which the boundary point between the two was traveling, the speed u. But I never told you what either one of them was separately, just that there is a relationship between the two of them. So the question is, as I change the various parameters, S0 of this Fisher-Kolmogorov equation and alpha that represents the coupling, what are the morphologies and what is the boundary that I have, let's say, between this morphology and this morphology? And what are the speeds in particular that we have over here? The speed in this case, we could get by this geometric construction. The speed here was kind of left unknown. So we did the simulation, and we found this rather surprising result to us initially, that the value of the speed when you are in any of these regions is completely independent of alpha, and given completely by the solution of this speed of the Fisher-Kolmogorov equation, as if this coupling term is not present. Okay? So there is a lot of work on the Fisher-Kolmogorov equation, telling us that the speed of the front of the Fisher-Kolmogorov equation depends on these two parameters, S0 and D, according to this square root combination. And what we find is that in one regime, we are given a speed completely by the Fisher equation, in one regime completely by this geometric construction. Yes.
So why is it that U has to be bigger than square root of 2 alpha?
Okay. So essentially this square root function. So basically, when we assume that we have a solution that is translating with some particular velocity, then the KPZ part of the equation on the left-hand side from d by dt gives you a slope. From the gradient square gives you a slope squared. So that's a quadratic equation, and that quadratic equation gives you this solution.
Yes, but in order for sigma to be real, you have to have 2 should be bigger than square root of 2 alpha beta. So I'm just asking that why is that necessary physically?
Okay, so in the other regime, the other solution is moving more rapidly. So the geometric construction gives you a speed, and you have to find the appropriate one of the 2 speeds. So essentially, it's just one equation by itself. The Fisher equation has a speed. The other equation, by this geometric construction, you can get a speed. It has nothing to do with the Fisher equation. It just depends on alpha. And you just have to compare the 2. And what we find is that the largest one of those is important. And that's what is showing here. So you essentially switch from one to the other. But the solution for the Fisher equation actually is more complicated if the function that we have for favoring one over the other itself depends on f. So this is really the preference of one over the other is itself something that depends on f. And this has a biological meaning, which says that essentially the species that corresponds to f equals to 1 is favored only if the concentration of f goes beyond a certain characteristic. So essentially, you need some neighbors that are like you in order to be able to compete and start to grow. This is called the Ali effect. And then in the language of Fisher-Kolmogorov equations, you move from the realm of pulled wave to pushed waves, and things become more complicated. And actually what we find is that the speed has a complicated dependence on various parameters, and it is also possible for the speed to become negative. So there is another regime in which essentially, because of the difference in the growth in the vertical direction, blue that was usually successful suddenly becomes unsuccessful. This is a little bit more mathematical that I like to give in my usual talks, but if there's any question, I'm happy to answer. Okay, this is maybe more fun. So one of the things that was mentioned today was this Kohl-Hopf transformation that converted the nonlinear KPZ equation to this stochastic heat equation. I'm not interested in the stochastic version, just on the deterministic version of this transformation when there is no noise. So I don't have to worry about existence of solution, things like that. I usually don't care about. So what this transformation allows us to do is, since I can solve W from some initial condition, I can calculate what the evolution of a height profile is in the absence of noise if I start from some initial condition. So the question that we asked is it possible to find a corresponding transformation that allows us to solve these 2 coupled equations that I had introduced for the fraction f that was satisfying the Fisher-Kolmogorov equation simultaneously with the height function that satisfies the KPZ equation. Okay, so for that, surprisingly, there is a connection to another type of model that is used in biological system. It's called the eigenmodel. The eigenmodel imagines that you have species that are indexed by alpha. It could be just 2. It could be more than 2 species. And each one of them grows exponentially with some growth rate G alpha. Now we allow these species to mutate between themselves. so that at some rate the species alpha can become species beta and vice versa. And we also allow these species to distribute themselves in space through diffusion. Okay? Now, this set of equations is very nice on one hand because they are linear equations as I have written. So there is some linear operator that I can start with some initial distribution of these species over space, and then through the exponential of this linear operator, I can find out what their number is at some later time. Now, the problem with this eigenmodel is that exponentials either grow to infinity or decay to 0. And in particular, I can, for example, look at the total number of all of these things by adding all of the n alphas. And I find that the total number diffuses but has a growth rate that represents the average of all of the growth rates, and it either exponentially grows or decays. And this average growth rate is obtained as a product of the growth rates of the different species and the frequencies of the different species. So I can ask, well, how does the frequency of species alpha, which is n alpha at point x divided by n at point x, how does that change? And this is the equation that governs that. Some of it is easy to see, like the growth of a fraction alpha is related to whether that growth rate is larger or smaller than the average growth rate. There is mutation between them, but the diffusion term is interesting because when I do the algebra of taking second derivatives and all of that, I find that the diffusion term involves a coupling of the gradient of frequency with the gradient of log n. Okay? Now, think back to this Kohlhoff transformation. I can define the gradient of log n as some kind of a height function, and then replace these equations in the language of what this height function is and what the growth of the different species is. And the height function essentially satisfies the KPZ equation, not surprisingly given the Kohlhoff transformation, but it has a growth rate that is reflection of the average growth rate that we had indicated. And the different species, the fractions of them, fractions are now between 0 and 1. There is no divergence. They grow according to the difference of the growth rate of the species from the average. And then there is a coupling between the gradient of the fraction and gradient of the height function. So we say, oh, I had seen these equations before. In fact, these are exactly the equations that I had introduced before, except that I have landed on a specific set of parameters, because previously I had some alpha here taken completely independent of everything else. But if these two equations originate from the same Kohlhoff transformation, I'm required to make these two parameters related in some particular fashion. Okay? Now, if I ask, if I go back to the phase diagram that I had, where do these special parameters take place? They're exactly at the boundary of going between one phase or to the other. So somehow this boundary line is something that is exactly solvable through this Kohlhoff transformation. And what it allows me to do, for example, is I can start with some initial condition in the height profile indicated with this lowest curve, and seed that initial profile with the two species, which I have indicated by the colors gray and dark brown. And I can solve the deterministic equations exactly and follow what the profile is. And what one finds is that the profile of the height function very quickly goes into these parabolas. If you take its gradient, you get these shocks that you would have in the Berger's equation. And what happens is that within each parabola, all of the bacteria are descendants of the highest point that led to that parabola. So all of these are really descendants of the bacteria that were over here. And the thing that is interesting about this picture is that actually this brown bacteria, I had given it less fitness compared to the gray. So that if I had stretched this thing as a straight line, the brown bacteria would have completely disappeared. But I placed them on a good location, and thanks to the location of their birth, they were able to survive and create their own niche. So it's good to be born at the right place. Any questions? Yes?
So just scroll up a bit. I don't have any intuition anymore. Yeah, so the top equation on your screen, do you have any intuition for this grad f dot grad log n? I mean, why shouldn't the dynamics and frequency care about the total population?
Okay. So mathematically, it comes from taking essentially the derivative of this equation. You get a term that is n alpha dot over n. You also get a term that is n dot over n squared. Essentially, the ability to sort of grow or diffuse really depends on how many other things are around you. And so the total number of other species that are around you modifies the ability to go in one direction or the other. And if there's too many things in one direction, you may want to avoid it.
Maybe a related question is, so you can call log n h, but just physically it doesn't really represent a height, right?
No. No. So basically this is important. There is one fictional model, the eigenmodel, that I can solve exactly, but doesn't correspond to anything because it has exponential growth. I do a mapping between that fictional model through this height function to another model, which corresponds to a height and species that are living on that height. And the fractions are going to be between 0 and 1. The height, which is the log of n, n was growing exponentially, the height grows linearly, which makes sense. So everything, so there is a fictional eigenmodel, and then there is a realistic growth on an interface model that is related to that. Okay? All right. So in the cases that I described, the species were immutable. They either had identical growth rates or different growth rates, but those growth rates were maintained over all times. But in reality, there are mutations, and because of mutations, there are fluctuations in fitness also. So far we didn't have mutations. And one extreme case of mutations is to imagine that somehow mutations will allow you to specialize in two different ways. For example, some trees may decide to become tall to take advantage of the sunlight. Others may decide to become broad. And so then the original population could potentially break down into two different populations. Now, inspired by Landau, Ginsberg, etc., we said, let's introduce some kind of a parameter, like an order parameter m, that distinguishes between being tall and being broad, or two different kinds of specialization. And let's assume that the fitness depends in some fashion on this order parameter. And again, I may want to maybe expand this in powers of m. It's the fact that there is some similarity to Landau-Ginzburg is incidental. It's just some function of m that I have analytically expanded. So then the story is that if I have two species that are next to each other in the kinds of model that we have been describing, it makes sense that I ask what are the fitnesses of the two species, and then I select the one or the other, the one that is more fitter to grow and have a child. So the mutations modify the fitness determines which one reproduces, and so we can sort of have some kind of a mutation in which the traits to become higher or become broader get modified through these mutational variations, and then this process continues. I start with a population that is focused on m equals to 0. It's unspecialized, and potentially as they accumulate more and more mutations, they may decide to split into two new species. So we looked at a number of different functional forms. One functional form that is not particularly useful for speciation is where the m equals to 0 or in vicinity of m equals to 0 is the most fit. And then the population really just goes on as a mixture and does not specialize. However, if I turn this around and in the vicinity of m equals to 0 is the least fit, and by specializing you can gain fitness. So these ones that have by mutation gone to m's that are positive or negative suddenly become comparatively more fit. And then we start with some kind of cloud of people that are generalists, and then as a function of time they accumulate mutations either on one branch or the other branch, and so you get these specialists that become more and more well defined, and they compete with each other, and ultimately one of them takes over. Another type of free energy landscape is one where locally m equals to 0 is fit, but globally specialization gives you advantage. And then in this case, you have to wait for a while until fluctuations get you from over this barrier, and then you have these explosive events in which one or the other of these species rapidly takes over. And finally, maybe more interesting, is it may be that m equals to 0 is unfit and you want to specialize, but there is so much fitness that you can gain through specialization. Again, you find a picture that is not too different from this picture if you look at it, because you essentially get things that are on one branch or the other branch, one maxima or the other maxima, and they compete with each other. The interesting thing is that if you ask how long does it take so that everything becomes one color, which is called fixation in biology, you find that the fixation time scales differently with the size of the boundary if you are in the regime where the fitness is bounded compared to the regime where the fitness can grow independently. And actually, the exponents that you get are precisely these dynamical exponents that correspond to z equals to 2 of the Edwards-Wilkinson model, or z equals to 3/2 of the KPZ model. And the reason for that is that you can think of a picture of the fitness, and if it is unbounded, the fitness keeps growing and growing, whereas in the other case, if it is bounded, it comes to rest, and the fluctuations become more or less like fluctuations around equilibrium. So it seems like by looking at distribution of fitness, we can gain some information potentially about what is going on in terms of the fitness landscape, whether it's bounded or unbounded or other things. So let me ask the question, if I have a bunch of individuals like us, we are not genetically identical because we have accumulated different set of mutations, and maybe because of that there's variations in various properties such as fitness. So what is the distribution? So one example that we can start to think about is what is called the mother machine that was developed in San Diego by Seok-Chun Jun, where essentially the bacteria are forced to grow in one column, and essentially at the front there is only room for one child and then the next child. And if we assume that each child gets some random mutation, since there is no competition going on, there is no reason why these fitnesses of the child compared to the parent should be positive or negative. So the best guess is at the end of the day, if I look at the fitnesses of all of the individuals at the top, they will be Gaussian distributed. It's sort of zeroth order assumption. I see no reason why that shouldn't be the case. But now suppose I go and do this with my model, where I have the two species, where their fitness because of the random mutations are different, but now the difference in fitness is important and only the better one survives. So I can write down an equation for how the fitness evolves in this case, which this is actually another one of those models that are in the KPZ universality class, and one can take a look at. And since we learned that the distribution of the height of the interface is governed by the Tracy-Widom universality class, I would expect that if I go and look at the experiment that I showed you at the beginning and fitnesses of all of the individuals that were at the top, and somehow measure them, that the distribution would not be Gaussian, would be Tracy-Widom. And again, the reason for that is that in competition, you lose things that are on the less fit side and you add them to the more fit side. You expect something that is a skewed distribution. And this is a nice skewed distribution. But one can ask more generally. So there is this nice experiment that has been going on for many years, where this long-term evolution experiment, essentially E. coli bacteria are grown and then diluted again, and over many, many generations, they have frozen the bacteria from past generation, etc., and asked the question, this long-term evolution experiment, how does the fitness change as a function of time? And this is a plot of the average fitness. It keeps growing, but it's kind of slowing down. Okay? So it's a different type of process that is going on. I don't know what is causing this, but my guess is that if one were to look at the distribution of fitness, as long as the fitness is increasing, there will be some kind of a skewed distribution. I don't think it will be Tracy-Widom because it's not a one-dimensional system, but it probably will be a skewed distribution. So recently we came across this suggestion that as you get more fit, in some cases the mutations that you make become less and less. So rather than changing some trait such as the height or width as a function of mutations, maybe one can change the rate of mutation itself as a function of fitness. And so Sergio recently looked at 3 different cases by which the rate of mutation is decreasing, found how the mean fitness changes, and calculated the distributions. And these are, again, skew distributions, but there's a lot of more work to be done to think about what are the different mechanisms by which species compete, fitnesses change, their distributions change, what are possible universality classes. So I think there's a lot of work that can be done in this direction. So in summary, we started with this nice experiment in which competing species that are neutral grow against each other, and by chance they form sectors, and the boundaries between these sectors satisfy this super diffusive behavior that can be characterized by the fluctuations of the boundary being governed by the dynamic fluctuations of the rough interface. Now, we also then went to the case where the species are not neutral, but compete with each other. And then the more interesting issue to consider is the kinds of morphologies that arise. And we saw a number of different morphologies that have some experimental character. And we have predicted some more morphologies, but I didn't go into them. And on the mathematical front, there is a question of characterizing the speed and characteristics of this coupled Fisher equation and KPZ equation. And there are some interesting behaviors that one can mathematically describe. The Kohl-Hopf transformation allows us to look at some deterministic limits and give us these nice pictures in which the initial condition can be solved exactly. And finally, that when the true mutations, the actual reproductive capacity or fitness of the species changes, one can have these different phenomena such as speciation, but possibly more interestingly, there are already universality classes characterized by whether the fitness can grow independently, indefinitely or slowed down, and there should be corresponding signatures in the distribution of fitness that has yet to be looked at. So this work was done with a nice group of students, Daniel Suartz, Lauren Lee, and Sergio, who's here, who recently joined this work. Kirill Korolov is a professor at Boston University. Hyun-Suk Lee did the experiments for these bacteria that show these V-shaped curves, and past colleagues include Jordan Horowitz, Dan Weller, David Nelson, who started a lot of this work on range expansion, and Sherry Chu, and I thank you for your attention.
Thanks. Yes. Yes, so with the V-shaped front, so if the competition between the blue and the black was more sort of intense, so if the blue started to eat then the black, or the black started to eat the blue, you would have sort of, instead of straight slopes, you could have some convex slopes. Have you looked at this?
We have not seen any case of that. There is so if you saw that in a lot of these cases, I kind of ignored the term Laplacian of H. Turns out that there are some cases where that Laplacian of H is very important. And they're like actually the one case that I showed you, there is not an initial linear growth, but there is a parabolic growth that starts, but eventually asymptotically they come to these shapes. So these are the most relevant terms. Irrelevant terms can give you corrections.
At least on the measurement there, the separation between the two interfaces is very linear. It doesn't seem that the fixation are no longer as relevant as you said at the beginning, when you could find some 1/3 or 2/3 fixation. So that there's still a little bit of fluctuation. So yes, you have something to say about it, or it's just so
yeah.
compared to the rest?
No, you're right. I mean, there's a number of interesting things to look at. I guess one of the things that I wanted to potentially focus on, I guess I can, yes. Okay, so you can see that right at the beginning, it doesn't start like a V shape. It grows like this, and then it goes. So, and over there, the fluctuations are also more visible. And so the answers that I have given are kind of in the asymptotic limit, where since things are scaling linearly with t, fluctuations that are of the order of t to the 1/3 have been ignored. So it would be interesting to include them, but at this stage we have not done so.
And at this level, you don't expect the answer collection or stuff like that into the company some sheet, or it is too tiny to see?
I think it is too tiny to see. There was, again, the case where I could solve things exactly. One could see that starting with some initial condition, there is a logarithmic approach to the final velocity. So I'm pretty sure that these Bramson corrections are there, but I don't think they have been looked at in the experimental system.
About the last part, is there a chance that the skewed distribution can be a realization of the characteristic distribution in higher dimensional APZ class?
I think it is a, so the question is, if one sees a skew distribution, what could it be? I would say it would be a characterization of, say, two-dimensional KPZ if the species are competing in 2 dimensions. Yeah. But in the experiment of this long-term evolution, they are completely mixed in a tube. So it may be an infinite dimensional version of this. So basically, I think there's a number of things that would go into characterizing the different universality classes. My guess is that the common element for all of them would be the skewness. And so maybe thinking about what the skewness is and...
Right. But people started to think about what kind of, maybe not distribution, but so there are a lot of discussions in the upper critical dimension.
Yes.
Class. And it could be that there is no finite upper critical dimension.
Yes.
And then people start to discuss what kind of distribution we may get at infinite dimension.
Right.
So I think there is some prediction on the skewness characteristics about that. Yeah. So interesting to compare.
I agree. Yes.
Thank you, Mehran, again.
Okay.